week 2 for the course on genes, genomes, and the epigenome. So, if at any point I'm not audible, please let me know. Um, just a quick recap. Hi, my name is Ankita. I will be your guide and the person who will be conducting lectures every week. We'll have sessions of around 2 hours, and we'll be having discussions ranging from basic biology, basic molecular biology, to current sequencing techniques, the updates on those, how advancement has occurred over the last couple of decades, and what is the current status, how you can analyze it, and how you can practically use it for research or for just for your own knowledge sake. So, in case you were not here for the last session for the last week, let me just quickly go over the topic. So, week 1, we focus majorly on the structure, function, and regulation of genes. Let me go into the presentation mode. We'll do a quick recap and then move into the discussion session for today. So, if at any point there's any doubt, any query, you can interrupt me and we can have a discussion on it immediately. And then we can move forward from that point onwards. So in the last session, we majorly we discussed the experiments that were done initially around 1868, around the late 1800s to the 1900s, where the chemical properties and the physical properties of the hereditary molecule for living organisms was being extensively studied. Majorly, we discussed experiments conducted by Frederick Miescher, McCarthy MacLeod, and Oswald T. Avery, where they performed different set of experiments to confirm that the nature of the molecule was indeed DNA and not the protein which was bound to it. And they confirmed that this was stable. It was able to pass information from one generation to the next and was indeed a landmark discovery in this field. Contributions were also made by Frederick Griffith, as we had mentioned, and from there, that point onwards, further confirmation was provided by another experiment conducted by Hershey and Chase, Alfred Hershey and Martha Chase, where they utilized bacteriophage, a type of virus, and they used radioactive isotopes for labeling the coat and the genetic material of the virus, and then observed that the material which actually gets transferred by the virus into a living cell, in this case, the living cell was a bacterial cell, they found that it was the DNA, the genetic material, and not the protein code which gets carried forward, thereby confirming the previously observed observations or the previously noted observations. And therefore, the discovery of DNA and its role had begun around this time. Moving forward, the structural characterization was carried out by Francis Crick and James Watson. It was very heavily aided by Rosalind Franklin as well, which we again discussed in the last session, where we talked about how she had performed X-ray crystallography and the results, which were results produced by her, were then further analyzed by these group of scientists, and finally they could decode the structure of DNA and the overall structure. Of course, we are familiar with it. It has a double helical structure of two antiparallel strands, which have complementarity between the bases. So this was the basic discovery which was carried out. And in 1962, James Watson, Francis Crick, and Maurice Wilkins were awarded the Nobel Prize for this. Rosalind Franklin's contribution was not acknowledged, considering that she had already passed on by that time, and posthumous awarding of the Nobel Prize had not begun at that point of time. Moving forward, we discuss the basic rules which are obeyed when DNA is actually formed. That involves Chargaff's rules, which majorly, if you briefly have to describe it, it involves the hydrogen bonding that exists between purine and pyrimidine bases in DNA, which gives it the stability that allows the hydrogen bonding to stabilize the helical structure of the molecule and maintain the fidelity of the information which is contained within the genome over generations. Apart from that, it also involved a few other rules, like the base composition varies from one species to another. However, from the same species, you would usually find a lot of similarity in the DNA content, and the physiological abundance of certain bases can also vary from one species to another. But the key contribution of Chargaff's rules involves this particular formula, which says A plus G equals T plus C, which is, of course, something that we understand at this point of time, since A, T, and G, C are hydrogen bonded in this manner, in this particular manner. This was further elaborated by Watson-Crick's model, whereby, based on Chargaff's rules, they could further conclude that indeed the twist, the helical twist that we find in DNA, is stabilized by this by these properties, and also a few other properties like the fact that it's a right-handed helix, the fact that the backbone is a phosphodiester backbone, and there are bases projecting towards the inside, which are hydrogen bonded to each other, all together combined to validate the information which had already been studied. Furthermore, the pairing of the two strands also created this major groove and the minor groove that we have already discussed, which allows the binding of transcription factors onto the DNA, which are able to read that information, decode it, and therefore regulate the control over how much of the gene expression should actually be allowed or disallowed. We discuss more on this later. So moving forward, we talked about the definition of a gene. It is a unit of heredity. Its position is fixed, or its locus is fixed within the entire genome, within a chromosome. And this entire structure that you're seeing, the schematic that you can see, represents how much of um, the amount of compaction that the genome undergoes for us to be able to visibly see it. And this is something we'll discuss in a lot more detail when we go into epigenetics, because there we'll understand the mechanistic insights into this compaction and the significance of having this compaction. But basically, we got an outline of what happens here. So you have 2 nanometer wide DNA undergoing extensive packaging by winding around a nucleosomal core, which is like a protein core around which DNA is wound. That allows the formation of nucleosomes, which then binds to histone H1 and creates a chromatosome. Then the chromatosomes also undergo further winding to form the 30 nanometer fiber in a solenoidal arrangement most of the time. And then that further forms loops. The loops then arrange into a fiber. The fibers arrange into the chromatid, and then two sister chromatids finally form the chromosome that we are able to see when we stain that too using a microscope. So the extent of compaction and the extent of information that is retained within a cell can be very strongly appreciated by this figure. What we also next discussed was that the information which is contained within the human genome in particular, of course, we are majorly concerned with that. We see that only 1.5% of the genome, which is a very small portion, is actually the protein coding part. The rest of it is majorly contributed to by LTRs or retrotransposons, lines, signs, and other miscellaneous heterochromatin. This was a very surprising discovery, contrary to what people had believed initially, that considering that we as humans, or for that matter, eukaryotes in general, have such an evolved genome that so much information is contained within our genomes, it was expected to be very largely protein coding, but surprisingly, we found the exact opposite results. This was further studied, which again is something we will also look into. But when this was further studied, we understood that just having information is not enough. Controlling the release of that information is a lot more significant, especially based on surrounding cues, based on the status that that particular cell is in at that point of time. So the environmental cues and the intracellular and intercellular cues also matter, and therefore the regulation of gene expression plays a huge role, and that has to be very tightly controlled and is extremely relevant in this case. Moving forward, we discussed how the architecture of a genome actually looks. So you can see that the central portion, which we are majorly concerned with, involves the open reading frame, or the ORF, which is actually the region which undergoes downstream processes like transcription and translation. There are upstream and downstream regulatory sequences, which are UTRs majorly. And apart from the UTRs, there are also several regions which help in increasing or decreasing the expression of a gene, majorly known as distal control elements or distal enhancers and distal silencers. So in a large number of cases, you'll also find enhancer and silencer written together, which again makes sense when we move further with this session, where you will see that the concept of insulators comes about, where they are able to insulate the information which is to be carried from one region of the genome to another. We'll discuss more on this. But basically, this is how the overall architecture of a genome, of a gene actually looks. And the central dogma of biology, when obeyed in the most simplistic manner, involves the conversion or the transcription of the DNA into mRNA and then its translation into protein. There are ifs and buts to this. We have discussed that as well in detail. But majorly, this is the default protocol that is followed by most protein coding genes. Moving forward, we talked about how gene function is regulated, and we saw how this again, I'm not going to details about this, but we discussed how much information is retained within one single chromosomal arm. We talked about promoters, enhancers, and boundary elements, how they are relevant to define a region of the genome, that if there is a boundary element that controls that only localized transcription will take place, while the rest of the genome will be left undisturbed. And there are enhancers which increase the expression of a gene, which increase either the recruitment of other controlling transcription factors, or by themselves they allow stronger transcription from a particular site. So there are various ways in which they can work. Next, we had looked at different steps at which eukaryotic gene expression can actually be controlled. So from each sub-step to another, you can see that from DNA to mRNA synthesis, transcriptional control and RNA processing control takes place. Once it has exited the nucleus and moved into the cytosol, RNA transport and localization control, as well as mRNA degradation, translation control, and protein activity control, all of these sub-steps, each of them can be switched on or off, or can be tuned down or um, paused depending upon what are the sensory cues that are being picked up from the environment. One major thing that we highlighted here was the fact that several post-translational modifications like phosphorylation, acetylation, and ubiquitination can occur at the last step, which actually controls the protein's activity. So protein synthesis has been completed, its folding has been carried out perfectly, and it is capable of being functional. But at the final step, its activity is again controlled by some modifications which are instilled upon that particular protein to control its activity. So that is, of course, something which we very regularly encounter when we study cell signaling, and we look at all of the phosphorylation and all of the simulation. All of that takes place and controls how much of a protein needs to be expressed, whether or not it at all needs to be expressed, whether it should be degraded, etc. All of that is communicated via these modifications. Um, then we looked at different types of control regions that are within the genome. So majorly, like we already talked about, enhancers and silencers which are present. So these are cis regulatory sequences, which mean that they are part of the genome itself. And there are other components which are not a part of the sequence, but that are able to control the expression of that sequence. And they are known as transfactors. Majorly, they are transcriptional regulators, coagulators, and proteins, basically, in general. So they actually are able to regulate the control of genes in a large number of cases. Example here was the fact that they are sometimes able to facilitate the looping of DNA. So why is that relevant at all? That is because there are certain regions which are usually quite far apart within the genome. For example, enhancers. They can be very far downstream in a large number of cases. So to bring the enhancer close to the region of interest to our promoters, sometimes mediator proteins, the one shown in purple here, is able to bring such regions close together and is able to control the expression that allows increased transcription of gene X, and thereby the overall bioavailability of that protein increases under those circumstances. So again, this is just an example. There are no like ifs and buts. There are no default cases that are expected. There are no default cases that are expected. There are only possibilities. This is the most frequent possibility, but there are again changes that are often in place. Um, This here we talked about regulation of gene function, and in this case we talked about how repression is controlled or is regulated within these cells under physiological conditions. Majorly we talked about 5 or 6 different methods by which this can occur. So either there can be competitive binding to DNA by an activator and a repressor. So both positive and negative signals can have a common binding motif, and they can compete with each other for binding to dictate how that gene's transcription will take place. Second, there is a possibility that the trans factors which bind to these sequences, they actually are able to physically um, sort of interfere with each other. They can mask the activation. So, sorry, activation surface and therefore prevent transcription from proceeding. Third, there can be direct interaction with GTFs so as to inhibit their activity. Like, for example, here the red colored repressor is able to bind to TF2D, which is a general transcription factor, and prevent it from doing its job. Fourth, you can see chromatin remodelers may be recruited, which are able to wrap up the chromosome once again and prevent transcription from proceeding locally in that region where it was previously supposed to do so. Then you have recruitment of histone deacetylases, which remove the acetyl group, remove the negative charge, and therefore they prevent the opening up of the chromatin, or they promote heterochromatinization and decreased transcription in that local site. Again, don't stress about this if you're not familiar with it. We'll discuss this in more detail in epigenetics. And the exact opposite can also occur where histone methyltransferases may be recruited. They might be able to bind proteins which are, they might be able to recruit proteins which are able to bind to methylated histones. And in those regions also, transcription is switched off. So these are all the different types in which methylation, or sorry, in which gene expression can be repressed by different proteins and different cofactors. Um, I think this was our, yeah, it was our final slide from the previous class where we talked about regulation of gene function by certain cis sequences like insulator elements, like you can see in here in blue, and some barrier sequences also, which are present. So barrier sequence here is shown in green, blue is insulator sequence. So insulator elements many a time can actually bind to each other. Again, this is doing a similar thing to what we have already discussed, where chromatin looping is facilitated by certain regions of the genome. So as a result of that loop, what is happening is that insulators are able to block the action of other sequences. And there are certain barrier sequences, like you can see, they prevent the spread of heterochromatin. So talking about the insulator proteins or the insulator sequences, why are they relevant? It is especially relevant so as to locally separate out a region of the genome. Again, why is that relevant? Say, for example, if you have an enhancer element which is located somewhere around this region. So this is the exact region where you want active transcription to proceed. However, very close to this region, there's another gene, say gene A in this case, which also can get regulated by the same enhancer elements. But that would not be desirable because that is not what the enhancer elements want. Cues are testing. So in this case, the binding of insulator elements to each other via an insulator binding protein can loop out this actively transcribed region, and therefore dictate specifically that this cis regulatory sequence is only going to transcribe gene B and not gene A. So that helps in removing any false transcription from proceeding in nearby locales. So this is another very major step in regulation. Apart from that, we ended on this note that from a single promoter for a particular gene, it's not just one simple signal that can dictate that yes, transcription is on, no, transcription is off. No, many a times there are multiple signals um, that are communicated to the same promoter, and an integration of all of those signals is taken into consideration before deciding whether or not transcription should be switched on or off, and whether or not it needs to be enhanced or repressed. All of these together can actually dictate the final fate of a particular gene and its expression inside a cell. Additionally, also keep in mind that it is very dynamic. It can change very quickly based on what is sensed somewhere around the cell, and accordingly fine tuning the expression of genes is very crucial and is very actively um, conducted within the cell at every single point of time within its life, within an individual, and as part of a system. So this is where we ended in the last class. We had discussed that we'll be moving forward and discussing how methylation also has a role to play in gene function, which is what we'll discuss first in today's class. And then we'll move on with the topic for week 2. So if there is any doubt, you can just let me know and then we'll proceed.
Ma'am, just like in gene A is not transcribed or uh, doesn't able to form protein, can we
Hmm.
also stop a defective part of gene that can't produce any defective protein which cause any problem in our body, just like a cancer and some cyst is formed due to some change in our gene, specific
Okay.
portion.
Right, I understood your point. But in that case, what would you want? You would want to introduce such insulator elements near such genes, near such defective genes, right? Is that what you're suggesting?
Yes, yes,
Okay. So, um, see, technically, it is possible if you're doing something like that in the lab, for example. But practically, why that will be a problem is because regions where such mutations are, which is going to be the topic for today, mutations, where defective proteins will be produced. Regions where such mutations will usually occur are regions which are already open. Why are they open? Because they are undergoing transcription. That is why the DNA is exposed, and that DNA is more susceptible to errors and more susceptible to get damaged. So in that case, you cannot it's not going to be the default fate of the cell to actually stop transcription in that region. Instead, what can be done is you can try to degrade that defective protein. You can use some tag or something which binds to the defective protein and remove that from the system to stop the disease from spreading further. But what you're saying, technically, it's possible if we synthesize, if we clone DNA, which actually carries such a defective protein, and put it between insulator elements and prevent it. But what you also have to keep in mind is that within a cell at any given point of time, there's not just one gene which is getting transcribed, right? So there are n number of other transcription complexes that are already assembled at different points, and there's always going to be a possibility that that defective gene is going to get transcribed by some region or the other. So it's a little difficult to predict how that is going to proceed, but what you're saying can be technically tried out. That's what I think.
because ma'am, in previous year the Nobel Prize
Hmm.
was given to a discovery for microRNA, which
Correct.
can behave as a switch. So can we
Correct.
use to inhibit that gene by microRNA?
Yeah. That is. If are you in academia currently, or you want to be?
I am currently in University of Delhi, and
Okay. So if you are familiar with, like, if you read papers or if you just have started off with it, you'll see that there are n number of papers where microRNAs are being studied because they have immense potential. So
yes,
they have their own, like, microRNAs are also not the end solution to everything. They
yes,
are a huge discovery in science, which is why it got that Nobel Prize, because it was unbelievable at that point of time. So their potential has been studied not just in cancer, but in other diseases as well. I think in some neurodegenerative diseases, in plants, they have been studied, but as different pvRNAs, etc. So non-coding RNAs by themselves have been used as targets, have been used as inhibitors And in fact, practically in labs also, like the way we work in the lab, we use siRNAs and shRNAs, which are short hairpin RNAs, to try and inhibit our gene of interest. So that is actively being studied and it is very promising. Where it is a challenge is that if you want to translate it onto bench for patients, there it becomes a challenge that how are you going to like feed people microRNA over an extended period of time, because an excess of something can also lead to non-specific results. Then you won't be able to control it. That if the microRNA goes and regulates some other region of the genome and that compromises some other function. So there people are currently stuck, but of course it is something that can change over time. We are not sure about it at the moment. But yeah, that is again an interesting field what you mentioned.
Okay, ma'am. Thank you.
Yeah. Yeah. Okay. My mic switching on. Yeah. For today's session, we are going to move a little forward. Majorly, we're going to discuss two topics today. If you go through the flyer, these were the sequential topics that we had listed out. So we are going to discuss a part of this today. And because I've gone into a lot of detail about mutation, we'll pick up polymorphisms and functional genomics in the next class. And then we'll move on to sequencing methods and more details about it. So the two major topics for today are A, how does methylation regulate gene expression? Because till now what I've mentioned are majorly about cis factors, right? Cis sequences that are undergoing different sort of modifications. And then we'll move on to mutations, their subtypes, their roles in disease, and why are they such a huge problem. So taking that forward, let us first understand the role of DNA methylation. What exactly do we mean? DNA methylation, as the name suggests, it is the addition of a methyl group, a CH3 group onto DNA, and it is considered to be an epigenetic modification of DNA, which has immense role to play not only in disease but in normal development as well. It includes transcription, embryonic development, genomic imprinting, genome stability, and chromatin structure. So this particular feature, genomic imprinting, it is something we will discuss, especially I think when we talk about epigenetics somewhere around the 6th or 7th week, because that's when I'll introduce epigenetics and then talk about genome imprinting and the immense studies that are being carried out about the diseases that get affected as part of it. So uh, basically, it has a large, very diverse set of roles to play. And the enzyme or the component within the cell which actually regulates it are the DNA methyltransferases. Obviously, as the name suggests, their substrate is DNA. Their job is to transfer the methyl group. Additionally, there are methyl CpG binding proteins and there are some chromatin remodeling factors as well. These remodelers, again, will pick up when we talk about epigenetics, but majorly for now, we can just keep this in mind that there are certain enzymes which are recruiting methyl groups onto certain regions of the DNA. Now, what does it actually look like if we look at it chemically? So here you can see cytosine, which is a normal or a canonical nucleotide which is present in DNA. And this actually contains, many a times can contain a methyl group where it looks like this, a methylated cytosine. Why is this relevant? It is especially relevant because within the entire chromosome, throughout not just humans but for any organism for that matter, any eukaryote for that matter, there are large stretches within the genome where these methylated cytosines are extremely abundant, and such regions are called as CPG islands, majorly because they consist of CG repeats, and these C residues or the cytosine residues are found to carry methyl groups attached on top of them. This is how it is usually represented. You will find this to be a common representation in multiple papers, multiple schematics, which indicates that this is a region which carries methylation, and that means that particular region is less accessible to the transcriptional machinery, or in other words, that region is transcriptionally silenced. We'll go into more detail about this. Don't worry. Firstly, let's go over the legend quickly and then we'll come to the figure. So, DNA methylation patterns are globally disrupted in cancer, with genome-wide hypomethylation and gene-specific hypermethylation events simultaneously occurring in the same cell. So please focus on the contradiction which exists in this statement. When we talk about genome-wide hypomethylation, it means that if you take the entire genome from a normal cell and from a cancerous cell, you can many a time see that the cancer cell overall has less methylation or hypomethylation. However, for certain specific genes, there is a preferential increase in methylation, which means that certain genes are silenced, whereas a large number of other genes are also found to express at very high levels. So there is a contradiction which is simultaneously existing, and it is very crucial and is something which is extensively being studied in today's time, that how DNA methylation patterns can be major drivers for the onset and progression and for worsening of diseases like cancer. So this is a very major event. We'll talk about it in more detail. But apart from that, there are also other diseases that are affected by DNA methylation. For example, here you can see that normal imprinting. Okay, let me just briefly explain what imprinting means. It basically means that the genome carries a mark of whether or not it should be immediately expressed or not expressed. That mark is basically a methylation mark. So when we talk about imprinting, it means that when after fertilization, when a particular zygote develops into an individual, they are carrying the information from both their parents, from the mother and from the father. The maternal and paternal alleles each carry a memory of the original parent that they came from, because obviously it is a gamete that was derived from that parent. So as a result, based on the information it has taken forward from the parent, it dictates how the zygote will progressively develop and which genes will be allowed to express or not express in the next generation. So basically, information from your father and your mother has been carried over to you, not just in terms of genes, but in fact, the mark on the gene is also being controlled. Now, which of the two alleles information will actually be executed or will actually be taken forward, that depends upon the region which you are studying. That depends upon what are the environmental conditions you are subject to, what is your diet, your lifestyle, a hell number of other things. So it is a fascinating area of research, which is genomic imprinting, and it is extensively studied and is very interesting. And now there are new arenas also about it, diet and lifestyle and mental health. All of these things can also have a huge role to play in this transgenerational genomic imprinting. Again, a fascinating area of research. We'll discuss it later on. But as you can understand, this is a process which is necessary for information to be properly conveyed from one generation to the next. When this is lost, it is often found to result in a lot of genetic diseases as well. Some examples I've cited here: Beckwith-Peidman, Prader-Willi syndrome, very commonly seen, not very commonly, but still quite prevalent in the Caucasian population. Angelman syndrome. These two are also part of the same locus. Again, something very interesting. We'll discuss it later on. Other syndromes as well, like Albright hereditary osteodystrophy, etc. So these are all diseases that are a result of improper information carried over in the DNA, which was supposed to be methylated in a particular individual. So these are just some examples. Now coming to the disease which we are most interested in overall in this course, which is cancer. So what you see here is just a representative region of the genomic DNA, which you expect to see in a normal cell. So what does it have? You have certain regions. Let me just read this legend. I think it will make it easier. Yeah, so you'll have some repeat rich regions which undergo hypermethylation. Hypermethylation simply means more number of methyl groups which are present on the CG repeats which are present in that region. So again, indicated by those sort of lollipop plots, which you can see here. And this is the pericentromeric heterochromatin, which means it is close to the centromere and it is heterochromatinized. And you can also see an actively transcribed tumor suppressor gene, which is TSG, that is associated with a hypomethylated CpG island. So this is, okay, again, another information to keep in your mind, that this represents methylation, but when the circle within this particular mark is colored, that indicates hypermethylation. When it is not colored, it means hypermethylation. This does not mean that there is no methylation in this site, it's just less. And here you can see these regions, it carries more methylation. So basically what happens is that there can be a sort of a balance that can be maintained within a normal cell between methylation and more and less methylation that overall controls the expression of a gene. Now, when this balance is disrupted, what it can do is that it can upset the control of expression in certain genes, and that can eventually lead to the onset of diseases such as cancer. Let us go to the next slide. I think it will help you understand this better. Yeah. So this is what you see for a normal cell. This is what you see for a tumor or a cancerous cell. So for a normal cell, you will see that tumor suppressor genes are majorly hypomethylated, less methylated, which means that increased expression or high expression of tumor suppressors are found. Why is that necessary? It is necessary to maintain the sanctity of the cell, prevent the neoplastic transformation and the development of cancer in such cells. Uh, Additionally, oncogenes, which are also normally part of the genome, they are kept hypermethylated to prevent their increased and inappropriate expression. In contrast, when you look at a tumor cell, you will see the reversal of these two phenotypes. You'll see addition of more and more methyl groups on the tumor suppressor, which means that the TSG is now silenced and the oncogene is now overexpressed. Again, keep in mind, this is not the only thing that happens. There are multiple abnormal signals which altogether are sort of there is a synchrony between all of these signals, which altogether leads to the onset and progression of cancer. But this is one major aspect which is now being discovered, not just for the sake of studying, but they are actually being used as biomarkers. So methylation of certain regions are being actually studied for liquid biopsy, where patient blood sample can be collected, and then within that blood, whatever fragmented DNA can be obtained from that patient's cancer cell, if that contains methylation of some tumor suppressor or hypermethylation of some tumor suppressor or the hypomethylation of some oncogene, then that can be used as a sort of a stratification uh, plan, whereby that particular patient will now be classified to a certain subtype, and accordingly the most optimal treatment for that patient can be used, and more detailed diagnostic and prognostic plans can be made. So it's a huge area of research and a very promising area of research which is being extensively studied in today's time. Now coming to the sort of model that shows how normal DNA methylation is actually disrupted in the cancer cell. So we'll look at both the schematic and the text to sort of maintain a tandem between the two. So what you see here on the top, at the top is the normal cell, and at the bottom is the cancerous cell. So here you can see normal cells, there is a low level transcription from hypermethylated repetitive DNA, which results in the generation of double-stranded RNAs that can be cleaved by dicer. This again, if since you're already familiar with non-coding RNA, you would also know the internal process that is followed for non-coding RNA downstream processing. This often results in the recruitment of repressive RNAi machinery and, in one way or another, DNA methylation and methyl CpG binding proteins. So DNMTs and other proteins can be recruited by the repressive RNAi machinery. So basically, this is what you see. This region undergoes very basal, very low level of transcription, produces dsRNAs, gets processed, recruits repressive machinery. Okay. Now, CpG islands, they are largely hypomethylated, and the associated genes, like a tumor suppressor gene, undergoes transcription in such a case. This is what you see here, that tumor suppressor gene is on, the cell is fine. Now, the binding of CCC-TC binding factor, or CTCF, also protects regions from DNA methylation, thereby keeping the transcription of the tumor suppressor gene on. Now, during the progression of a cell towards a cancerous state, that now the cell is undergoing neoplastic transformation, then what is happening? You can see that the DNA tends, the repetitive DNA, it tends to lose methylation marks. Majorly, there can be due to some DNMT dysregulation or the lack of double-stranded RNA processing, basically loss of control. What that does is it increases genomic instability. What do we mean by instability? It means that regions which were supposed to be silenced are now getting randomly transcribed. So there is information that should not have been there and can now manipulate all the existing information and therefore confuse the cell and push it towards a state where it just becomes out of control and favors signals which promote its survival, promote its proliferation, and thereby eventually progress towards cancer. So, repeat hypermethylation near genes or promoters could disrupt the transcription of TSGs by several mechanisms, including transcriptional interference, generation of antisense RNA, recruitment of RNAi machinery, etc. In addition, the re-expression of Boris might also displace CTCF from its binding site and disrupt the methylation boundary functions of CTCF. And now DNMTs can gain access to CPG islands. You don't have to get into the technicalities, but basically what happens is that when this balance is upset due to some DNMT mutation or for whatever reasons, there is a loss of control on the hyper and hypomethylated regions within the genome, then there can be a reversal of phenotype and preferential silencing of necessary genes can occur, and unwanted genes can now undergo very high expression, thereby allowing the development of cancer. So this is one thing, one major shift that happens, that the balance between hyper and hypomethylation is now lost. Now, I think I just Now, what is the outcome of this? The outcome of this is that methylation of the DNA can now dictate whether or not a cell will go or not go into cancer. Also, keep in mind that this is not the end of the fate of the cell. There can be further recruitment, there can be chromatin remodelers which can come in. There can be heterochromatinization, but again, keep in mind that will be much less prevalent once there are a multitude of signals that are no longer able to protect the cell will almost always move into a condition where it is beyond control and it is now going to become cancerous. And therefore, detecting early signals like early dictators of possible incorrect DNA methylation has become a relevant biomarker that is being clinically used to diagnose patients as early as possible before it has progressed to a stage where they can no longer be treated. So this is something, this is a new area of research. It's very promising and it's very interesting. And we'll learn more about it, not directly about DNA methylation, but about methylation and the epigenome when we discuss the epigenetics part of this course. So this is basically where I wanted to end gene regulation as a continuation of the previous class. Again, if there are any doubts, please interrupt me and let me know. Uh, we will next move on to the next set of topics for today, which are mutations. So again, this is not a very new topic, but the reason why I wanted to go over this in complete detail is to make sure A, that you have brushed up all your basics, B, you don't have any confusion between the types and subtypes of mutations. Why? This is especially relevant because we will discuss next generation sequencing techniques in the upcoming weeks, and a major use of these NGS technologies in context of people, in context of translation, is the fact that people are now trying to detect what sort of mutations can actually dictate the course of a disease and whether or not it can define the subtypes of people within a population towards having certain tendencies, whether they are carrying certain marks which are, classifying the disease further, a number of other things. So mutations become a very, very major part of high throughput sequencing technologies in today's time. So before we go too far, the best idea is always to clear your basics. So that's what we're going to do today with some new information as well. Now, for your reference, most of the information that I will be presenting in today's class has been taken from Russell iGenetics. You can refer to any textbook of your choice, anything which you are already familiar with, if it's a part of your coursework or whatever is most accessible to you. Additionally, a lot of information has just been taken from Google Images, whatever conveys the information most easy. You can use any online resource. You can let me know if you need anything. I'll try to look up for more better resources that you can study from. So with that background, basically we are going to talk about mutations. Now, despite the near perfect fidelity of genetic replication, infrequent unrepaired mistakes in the replication process can often lead to changes in the nucleotide sequence of DNA, producing a genetic mutation. What is it? It is a change. Very simple definition is that it is a change in the nucleotide sequence of a short region within the genome. So it's simply a change in the nucleotide sequence. That's all it is. That's the most compact, most accurate description of a mutation. Of course, there are many adjectives that you can add on it, many circumstantial definitions that you can add on to it. The most basic definition is exactly this. Now, the question is, how do mutations arise? Again, this is something that I've just introduced here. We have gone into more detail about this in later slides. But mutations majorly occur because of two reasons. First, some mutations are just spontaneous errors. They occur majorly during the replication process, and they are basically able to evade the proofreading ability of DNA polymerases that synthesize polynucleotides at the replication fork. So these are very commonly often also known as polymerase errors. That is because in the eukaryotic system, the process of replication is so finely controlled. If you studied replication, you would understand that there are multiple checkpoints. There are so many enzymes, so many proteins that are carrying out each and every sub-step. And a major quality control step is the enzyme which actually carries out the job of uh, polymerization. So basically, its job is to read the template and insert the correct nucleotide in response to it, which is complementary to it, right? So when there is an error in this process, in A, reading the correct base, second, recruiting the correct complementary base, and C, ensuring that it is forming the proper type of hydrogen bonds which are expected in a normal, regular form of DNA double helix. Any changes within this basic process is basically going to give rise to a mutation. Sorry. Now, these mutations are considered to be spontaneous, sorry, considered to be happening spontaneous because they are able to evade this proofreading function. But there is another set of mutations that are called as induced mutations. Why induced? That is because there is some external agent which has come in and brought this mutation within our gene sequence of interest. Most commonly, it is known as a mutagen. What is a mutagen? It is any agent, whether chemical, whether natural, any sort of agent, which is able to change the parent DNA, causing a structural change or a structural disruption that affects the base pairing capacity of the nucleotide. So this is an example of what an induced mutation kind of looks like. So before you can see, this is the regular double helix of the DNA. When there is incoming UV radiation, many a times it can lead to some sort of a disruption, as a result of which, instead of forming complementary bases with the complementary hydrogen bonds, there is some change, and instead of interstrand hydrogen bonding, there is intrastrand hydrogen bonding, and that is going to completely ruin the structure of the double helix. So this is again something very commonly seen, and it is something that does happen, and the reason it happens is something we'll discuss in a couple of slides. But apart from this, we also have another representation of what exactly happens during DNA replication and how mutation affects the same. So here you have a parent DNA molecule which undergoes DNA replication, for which both of these strands are separated, and in in complementarity, the daughter strand is synthesized, taking the first strand as the template. When there is a replication error, let me zoom in a little bit here. Yeah. When there's a replication error, if you see here in blue, then what it effectively does is that it now changes the template for the next generation. As a result, while the original base pairing involved T and A hydrogen bonding, the first DNA, you can see that a replication error has created a C and A bond. The consequence of this is that the next round of replication used as template, and in Taking that as the template, a G will be installed at that site. So a subsection of the DNA present within this particular individual will carry this misinformation across generations, and it is something which will then be carried forward in every subsequent cell. So this becomes a major issue and is something that we have been studying for a very long time. Now, somebody might ask that what is the big deal? If there is a base change, how does it really change anything? Now, to brush up your basics again, if you remember, when we talk about the central dogma, we understood how DNA regulates RNA and RNA regulates protein. Um, when I say regulates, I mean that it dictates the information which will be translated from DNA to RNA to protein. Now, a major catalytic step, so to say, in this process is the genetic code, which is obeyed universally. And this actually involves reading the information present within the mRNA and translate that into the correct protein. So why is that such a big challenge? The reason it's a big challenge is because there are so many possible combinations. As you can see, 64 possible combinations. Let me just get the 64 possible combination of triplet nucleotides, which means three nucleotides in tandem, that can possibly exist for each region of the genome. Now, among these, there are certain properties which have been standardized and published and accepted over decades now. Is the fact that A, the triplet codon or the genetic code is sorry, I have a bad throat. Um, the first property is that it is degenerate. What does degenerate means? It means that there is not just one amino acid. Sorry, there's not just one codon, triplet codon that can be used to dictate the presence of one amino acid. Obviously, that can be very well appreciated if you look at this genetic code. Let's take the example of valine, for example. Valine can be possibly encoded by all these four triplets. As you can see, there is a lot of similarity between them, that having G in the first position and T in the first position, and then a combination of any other base in the third position is going to uh, undoubtedly going to encode for valine. So that indicates the degeneracy of the genetic code. Second, the fact that it is non-overlapping. So in tandem, when there are multiple sequences, this you can appreciate by the figure which is at the bottom. It is continuous and is non-overlapping, which means that the ribosome, when it reads this particular sequence, it reads three bases and moves forward. It doesn't go back or backtrack and then read again from the second base and then again from the third base. No, so it is read in a continuous manner where the three bases are non-overlapping in nature. Third, it is comma-less, which means that there are no boundary elements or no punctuation marks which are present between different codons. Fourth, it is non-ambiguous. What does non-ambiguous means? It means that while a single amino acid can be encoded by multiple codons, a single codon only encodes for one amino acid. So, when an mRNA is being read, the ribosome should ideally never get confused as to whether this codon encodes for amino acid X or amino acid Y. There is no discrepancy regarding the same. However, the corollary is possible, whereby if for a particular protein we want to insert valine at a particular site, then the possibility of bases that can nucleotide bases or nucleotide moieties that can exist at that position can be variable, and there are four possibilities that can exist for the same. So that indicates the non-ambiguity of the genetic code. Finally, the code is universal in nature. It is nearly the same for all living organisms, thereby making it a very relevant area of study. So this is the overall recap of the genetic code, and the reason why it mattered, like we were discussing, is the fact that if there is a single base mutation, a single nucleotide mutation at a particular site, then that can dictate how that particular mRNA is going to be translated and which protein or which amino acid that particular region of the mRNA is going to encode. So based on that, it will dictate whether or not that protein is functional, what job it will do, whether it is a nascent protein or a mutant protein, like you were just discussing, whether it is going to be a defective molecule. All of that is very largely dictated by the genetic code because it does not change while DNA keeps on changing. So that is basically what ruins everything, so to say. So, yeah, so these are the different properties of the genetic code. And based on this, we will next discuss the type of mutations that can exist, which are actually impacted by the variations that can be seen. So, commonly, the types of mutations, you will find many different ways in which this is actually structured. So, sorry. So, the first subtype includes point mutations. Just give me a moment. Okay. So the first subtype includes point mutations. So a point mutation, as the name suggests, is literally a single change that occurs within the genome. If we look at the definition, it says that there is a change in a single nucleotide base in the DNA sequence. They can be further classified into many different subtypes. Now, what are the subtypes? The first one is a silent mutation. Second, a missense mutation. And third, a nonsense mutation. Let's understand them one by one. A silent mutation, let's look at the example also. A silent mutation, as the name suggests, it's a mutation that has no effect. It doesn't make any noise. Why so? This actually takes the example that I already mentioned, that if for a particular amino acid, there's a lot of degeneracy, which means that there are multiple possible codons that can encode for the same amino acid, and there's a switch from one nucleotide base to another, which is able to still is able to encode for the same amino acid, then in that case, that particular mutation is going to be silent. So here, even if there is a single base change, there is no effect, the protein works fine, it is functional, and therefore there is no change. However, there does exist one possibility there does exist one possibility that if that particular region also has some regulatory effect, then that is very heavily dependent upon what is the sequence of that region. Then in that case, that mutation can have an effect. Say, for example, you have a transcription factor which binds to a certain region of the genome, which is going to act as a regulatory cis sequence. And when that transcription factor binds, it uses a certain motif as the recognition sequence for binding. Then in that case, there can be some error, there can be less binding, there can be less binding, or there can be no binding at all in such cases, and therefore that is going to affect gene expression. Sorry, everybody has a sore throat. So this is the first example. Second is of a missense mutation. Now this is the most common, most relevant, and most clinically used subtype of mutation. When I say used, I mean it is of most interest. So missense mutation, as the name suggests, is something which is going to completely change the meaning of the original information, and it is going to stop making sense. So a change in the nucleotide that is going to result in a different amino acid being produced, and as a result of completely different protein also being produced, is actually going to be classified as a missense mutation. So the example here, again, let's go over it. If you see from serine, valine, proline, tyrosine, we're seeing that proline has now changed to threonine because of the change of a single cytosine into an adenosine. And as a result, it is going to completely change the properties of that protein. Now, again, keep in mind the fact that not every um, not every amino acid change is going to have an equal impact. Why? Certain amino acids are often also known to be special amino acids. Proline is one such example. It has a very distinctive structure. It has a ring that is part of the amino acid by itself, which is often considered as a helix breaker, and basically it's a disruptive structure. So its presence has some relevance within the protein sequence, and substitution of that particular amino acid by another one which has a more which can either have a completely different property, like for example threonine in this case, that can have a completely different property, or if there was a cytosine for sorry, if there was a cysteine, for example, that would have a completely different property. So the extent to which a particular missense mutation can have an impact within a protein can also vary. So that was just additional information I wanted you to have. In a in addition, there is another type of mutation where a protein just stops making sense and um, stops working also. In that case, it is called as a nonsense mutation. So what happens there is that there's a change in the nucleotide that converts a functional codon into a stop codon, leading to premature termination of protein synthesis. So the fact that any coding sequence can get abruptly stopped by the change of a single amino acid is something that's very commonly seen, and it is commonly known as a truncating mutation or a nonsense mutation. What it does is that it leads to a partially synthesized protein, which is more often than not non-functional, and especially it is extremely it's a waste of resources if it occurs very early on. And if it occurs after a partial part of the protein is synthesized, then that can also be equally dangerous, that it can have some aberrant effects which we don't even know about, and it can go and partly function or change the function of a known protein and can have very disastrous effects. So again, this is something that, if we go back to the genetic code, yeah, you will see that there are three stop codons that are recognized by the eukaryotic ribosome, TAA or UAA for that matter in the mRNA. UAA, UAG, and UGA, these are the three stop codons which are recognized by the ribosome, and that that if encountered are going to lead to immediate termination of the translation process, and therefore a partial protein will be synthesized. So this is how the this is how it looks. So you have this normal protein coding gene undergoing transcription and translation, and when there is a sense codon present like AAG, then that is going to allow translation to proceed, because a charged tRNA is going to bind, and then the polypeptide will be transferred from the previous mRNA to the next mRNA. I think from the P site to the A site, if I'm not wrong. These are the three sites within the ribosome, E, P, and A, or yeah, EP and A. And once the polypeptide has been transferred here, it is going to continue translation and the complete protein is going to be synthesized. In contrast, when there is a mutation of this sort where a T is changed to A, then that A is going to undergo transcription and translation, and that is going to be now encoding for a nonsense codon. Then that is going to recruit a release factor instead of a charged tRNA, as a result of which the release factor is going to prematurely terminate the translation process, and an incomplete polypeptide is going to be synthesized. This is basically what we discussed. Additionally, this is another example of what happens. I think I've kept it later on as well. Just give me a second. Yeah, I think I've kept it later as well. But anyway, let's discuss it here too. So this is an example of how replication errors can enrich for certain mistakes. Like for example, here you have the parental DNA, which is going to undergo DNA replication. And at the replication fork, you can see there is a separation of the two strands. And from the two template strands, now new daughter strands will be synthesized. Now, at this point of time, if there is an error that occurs whereby after separation, this particular guanine, instead of pairing with cytosine, takes a different route and it pairs with T, which is thymine, then in the next generation, the mismatched GD pair is again retained and it undergoes replication. Then in the next generation, which is the second generation, sorry, in the second generation, there is a complete change in the information which is carried forward. So what that can possibly do is that it is going to lead to a mutant phenotype for the same gene in the second generation. So there has been a GC to AT transition that has occurred after replication has been completed. So this is how replication errors are taken forward. Now we were talking about the types of mutations. We talked about the different types of point mutations. And there's a second subtype, very commonly and very prevalently studied and very relevant for diseases as well, which is known as frame shift mutation. What does it mean? It basically involves a class of mutations known as indels or insertions and deletions. So they, as the name literally suggests, they are involved in the addition of a certain base or the removal or the deletion of a certain base from a DNA sequence. So among these, you can find two indels, or among insertion deletion mutations, you can find two subtypes, the frame shift mutations and the in-frame mutations. So what are frame shift mutations, which is what we were looking at? Basically, it leads to the shift in the reading frame of the ribosome that it was originally following. So what you can see here involves A, G, C, G, T, A, and then triple C, and then TAC. So when it is read in its default format, it encodes for these four amino acids. However, when there is a deletion after CG, you can see three C's which are present here. So there has been a deletion of T and A. Then it leads to a complete change in the sequence. And instead of proline being encoded, you can see valine and leucine being encoded from that particular site. So this is a shift in the frame leading to the production of a completely different protein. This can occur either there can be insertion or deletion. This was an example of deletion. But in certain cases, there can be some insertion as well. For example, here you can see a single nucleotide insertion takes place. A G is inserted in between this sequence. Then that leads to complete shift of frame. And after methionine, instead of proline, tyrosine, and all these amino acids, you have arginine, valine, etc. being encoded. When there's a double nucleotide insertion, that can do the same as well. However, if there is an insertion of three nucleotides, which of course is much less prevalent, it is a large error for the polymerase to make. But in case that does happen and there are three bases that are inserted, then in that case, what happens? There is no effective shift in the frame because for example, imagine that after AUG, another CCG is inserted, then it will basically keep on reading the mRNA in the same frame, in the correct frame. But what will happen is that an extra amino acid will be incorporated within the polypeptide sequence, and the same thing can occur for deletion as well, where there can be deletion of a single nucleotide, of two nucleotides, or three nucleotides. When there's a deletion of three nucleotides, it does not shift the reading frame, but it leads to the loss of a single amino acid within the protein sequence, which again can potentially affect protein function and stop it from functioning completely as well. So again, these are context specific things. All possibilities exist. It's just that some things are more prevalent than others, but you would be very surprised to see how stats actually do favor such frame shift deletions and insertions in diseases like cancers, which are very like genetically heterogeneous. When we say heterogeneous, you'll find such huge range of alterations within the genome that is present in cancer cells. There are mutations, there are structural variations, chromosomal aberrations, there can be frame shift mutations, a lot of different things that can occur, and all of them are eventually leading to cancer. So it's a huge field of research and seems almost indefinite, but that's what keeps it interesting as well. So that is what I wanted to tell you about the types of mutations. Now, apart from the classification which I just mentioned, this is another way in which classification is also done. All these images, they have been taken from Russell I Genetics. It's a wonderful book. You can refer to it, not just for studying genetics, but for studying anything about the DNA for that matter. You'll find the PDF online. You can refer to it. This has been taken from the chapter about DNA mutations and DNA repair. So what you see here is a different way of classifying these already discussed topics. So you have transitions and transversions. Transition basically involves an AT to GC conversion, which means that there is a purine to purine change. And here you can see that while the original sequence had AT, instead of that, you will find a change. You'll find a change. So there's a purine to pyrimidine change. So from AT, you'll find there's a GC base pair which has come into place. On the other hand, when there's a transversion, then you'll find a similar change that has occurred. That from a CG, you find a GC base pair to be present at the same locus. However, this is not as innocent as it looks. It's not just a simple base change. Imagine the fact that within each strand of the DNA, the information has undergone a complete change. That is because what was previously CAA is now going to be read as GAA, and that is going to encode for a completely different amino acid. And in this case, the change has occurred on both strands of the DNA. So the potential of incorrect transcription and translation is even more, and therefore the errors can be even can be much worse and can be much more serious, and therefore transition and transversion mutations should also not be taken lightly, and they can wreak complete havoc within the genome, like I've titled this slide as well. Apart from that, you have missense mutations also. Sorry, you have missense mutations also, which change one amino acid to another, like we just mentioned, and that can change the codon from lysine to glutamic acid. So here you can see from a lysine, glutamic acid is being encoded. There are nonsense mutations where it abruptly terminates the translation process. So you can see from AT, you have TA switch, and that encodes for UAA codon, which is a stop codon, and terminates the translation process. Then you have a neutral mutation. This is a new terminology that we're coming across. Neutral mutations are a slightly vague classification of this subtype, whereby you have the change from one amino acid to another amino acid that has similar chemical properties. So what happens there is that it's effectively a type of missense mutation only because it is changing the amino acid. But why it is called neutral is because the impact of that change is not as immense as it would have been if the amino acids were completely different to each other. So, like we saw in the previous example, a proline to glycine switch is huge. But for example, an aspartic acid to glutamic acid switch is not that impactful. And they can, to some extent, conserve the properties of that protein. But of course, if it's a sequence specific activity that must take place, that can get severely impacted. So that's what a neutral mutation is understood as. In this example, they've shown lysine lysine to arginine switch. So they also have similar properties. So to some extent, the mutation, while is indeed a change in the genome, the effect of that mutation is not immense in comparison to what it could have been. Then comes a silent mutation. The difference between these two terminologies should be very sorted in your head, that a neutral mutation and a silent mutation are completely different. A silent mutation is one where the same amino acid gets specified, like we already discussed. Like for valine, there are four possible amino acids because of the degeneracy in the genome. So that change in the genome where the same amino acid is still getting encoded is a silent mutation. The mutation where a similar amino acid but yet a different one, different to the original one, is getting recruited is going to lead to a neutral mutation. Then comes frame shift. We have already talked about it. Here you can see that there's an insertion of a GC base pair, which completely messes up the information after glutamine and leads to a completely different polypeptide getting synthesized. So that is going to lead to a defective protein. So till here, if there are any doubts, you can let me know, and then we'll move on to other mutagens. So anything you want to discuss, you can let me know. Okay, if not, we'll move forward. So The next question that was to be addressed is that how do these mutations arise? We talked about one possibility, which is DNA replication. So, a little more detail about the same. This actually DNA replication errors. A, of course, are they occur in a situation where the polymerase's proofreading capacity is either compromised or is bypassed or is not properly taken care of. In that case, what happens is that there is a base pair substitution mutation which is occurring. That means one base pair to another change is occurring, and that occurs if mismatched base pairs are formed during DNA replication. How that happens, we'll come to that. But this is basically what it possibly looks like. So you have your template strand, and in response, or taking that as the template, a new daughter strand is synthesized. Now, what you see here is that there can often be the looping out of a single base within the template strand, either in the in the daughter or in the template. So here you can see that within the template, an A gets looped out, whereas in this case, a T from the newly synthesized strand is now looped out. As a consequence of this, what you see, sorry, as a consequence of this, when these two daughter strands are further producing complete DNA molecules and that is passed on to the next generation in the progeny, you will see that that particular base gets inserted. It does not get properly base paired, but when there is a separation of these or the denaturation of these two DNA molecules, not even denaturation, when there's a separation within the replication fork, the template that will now be formed will be completely different, and then that will recruit, this A will recruit a T, or this T will recruit an A, and therefore this mutation will be carried forward. So this is how DNA replication errors arise, and they are classified under the umbrella of spontaneous mutations. Now the fact that this occurs, and this is actually very prevalent, and to an extent also, I wouldn't say stable, but they persist within the genome. This actually occurs because of some unconventional or some non-canonical base pairing that occurs. So chemically, if you see here, each base can exist in different states, different conformations, which are known as tautomers. Now, when a base changes its state, what do we mean by state? There's a keto form and there's an enol form. So when DNA, when you look at physiological DNA, most of the time these bases they're found in their keto form, and that actually allows this normal Watson-Crick base pairing to occur, where T binds to, or A binds to T and G binds to C. So this is what you see here. This is the normal Watson-Crick base pairing that occurs between A, T and A, and between C and G. You can see two bonds and three bonds that take place. However, when there is a sort of when there's a shift, a tautomeric shift, that means the base has changed its state. There you can see unconventional forms of base pairing does occur. So this is your normal thymine, and this is your rare form or rare enol form of guanine. There, T and G are able to form base pairs, and they form three base pairs. Similarly, normal cytosine and rare imino form of adenine, they are able to form two base pairs. So ATGC, or the sanctity of ATGC base pairing, which is obeyed by or which is dictated by Shargav's rule, is no longer followed. And instead, what you see are very abnormal base pairs, which are not very stable as well, which involves thymine and guanine, or TG and AC base pairing to now occur. So this is actually a major problem that is very commonly seen. Apart from that, Apart from that, you also have spontaneous chemical changes, which include depurination and deamination of certain bases. These are chemical events. Do not confuse the two. The first one that we talked about was about DNA replication errors. And as a subtype of that, we talked about the fact that there are base pair substitutions which take place, which can be attributed to the change in the state of a particular base or the conformation of a particular nucleotide base within the sequence. As a result of that, incorrect base incorporation takes place. What we are talking about now involves chemical changes. So among these chemical changes, we have two examples. We have depurination and deamination. What it effectively does is that it creates lesions. What are lesions? Lesions are damaged sites within the DNA. So what it effectively does is that it compromises or it affects the fidelity or the correct information or the sanctity of the genome. Why is that a problem? That is because while we think about it, that a somatic mutation is not really that big a problem. If, for example, um, say my nail bed, cells in my nail bed, they're dividing, and one of these cells is carrying the huge mutation which is leading to some immense change. Yes, that is going to be a problem, but that's going to be a very small problem. Errors in critical organs, and when those errors are propagated over multiple generations, can lead to neoplastic transformation, can lead to the formation of small masses, bigger masses can spread, become metastatic, and can become a huge problem that is, in a large number of cases, untreatable and almost lethal. So, understanding the origin of these errors and trying to troubleshoot them is something that we always try to do. And two of these errors, which can be introduced by some mutagens, which are spontaneous. The nature in which these mutagens can work is spontaneous, therefore, calling them spontaneous chemical changes involves these two types of modifications. The first one, which I just mentioned, is depurination. As the name suggests, there is a removal of a purine group from DNA, or yeah, just an empty site in that region. So it is the loss of a purine from DNA when the bond hydrolyzes between the base and the deoxyribose sugar, resulting in the generation of an abasic or an apurinic site. It's just an empty site within the genome. Why does it occur? It occurs because the covalent bond between the sugar and the purine is much less stable than the bond between the sugar and the pyrimidine, and therefore the purine bond is much more prone to breakage. Therefore, we never come or we rarely come across something called as a deep pyrimidination. That doesn't happen. Purines are much easier to break, and therefore they are much more prevalent. The other possibility is the presence of or the removal of an amine group by a process known as deamination. And this occurs usually in context of a nucleotide base. Why at all? That is because the deamination of cytosine under normal circumstances or physiologically within the cell produces uracil, which is a normal component of RNA, but is never ever present under normal circumstances within DNA. So the presence of such deaminated sites can also change the fidelity of the genome. How so? For example, if you have cytosine which undergoes deamination, it is producing uracil. Um, there are certain mechanisms that are in place within cells. Majorly, it is BER or base excision repair, which is able to recognize the presence of such erroneous bases and is able to discard them or remove them. However, however, when there is deamination of 5-methyl cytosine, that is 5-MC, what it effectively becomes is thymine. So a region which previously contained cytosine has undergone methylation at the 5' site and has also undergone deamination. Both these processes have now converted it into a completely different nucleotide base, which is thymine. And what is interesting in this case is that because it is a nascent component of human DNA, it will not even be recognized as an error. It will get integrated and propagated across generations, thereby completely changing the information which was previously encoded within that region of the genome. So deamination of a canonical base, while it has some effect, deamination of a non-canonical nucleotide base has much more adverse effects. So that is another form in which spontaneous chemical changes can become a problem. Another source of such spontaneous changes includes radiation. In this case, radiation, the one we have discussed, is UV light. There is also ionizing radiations, of course, that causes breaks within the DNA. And when I say break, it basically means it fragments DNA. And in such cases, DNA repair also takes place. There are There's homologous DNA repair that occurs in that case, or non-homologous end joining, where there are double strand or single strand breaks, where it tries to look for regions where there are where there's microhomology and tries to align such sites and then tries to stitch together the broken DNA to make it continuous. So that's a completely different area of ionizing radiation. I've not gone into details about it. But apart from that, non-ionizing radiation is also a source of mutagens. The most prevalent example in such cases is the UV light. So, UV light or ultraviolet light, what it does is it causes mutations by increasing the chemical energy of certain molecules like pyrimidines in DNA. So, one effect of UV radiation is the formation of these abnormal chemical bonds, most commonly between adjacent pyrimidines, particularly between two consecutive thymines, and they are more often than not present in one strand. So, you have two parallel strands, you expect these two hydrogen bond to each other. However, when within a strand there is an intra-strand base pairing between two subsequent bases, what that is going to do is it's going to leave the other side open, prone to nucleotide damage, prone to restriction digestion, prone to n number of defects. So therefore, in that case, what becomes relevant is the fact that you have to protect the genome and you have to look at or you have to avoid exposure to these kind of things. Again, there is there are repair mechanisms in place that are able to remove these moieties known as DNA adducts. Adducts because they form these large um, interruptions within the genome. And when such thymine dimers are also formed, what you also need to understand is that replication is going to be interrupted because the replication fork will not be able to move forward in such a region. So there are repair mechanisms which are also able to remove these also in due time. However, when there is a huge accumulation of such DNA adducts throughout the genome, then it becomes a problem. In that case, it becomes challenging to resolve such a problem, and then aberrant and uncontrolled and incorrect replication will start taking place from other regions, and that is, of course, going to help the development of cancer. So yeah, that's what you see here, the formation of a thymine dimer. There are two adjacent thymine residues, the exposure to UV light, and the formation of this structure where it forms where there is an alignment of the two um, aromatic rings. Uh, not aromatic, but yeah, of the two planar rings. And then that leads to the formation of the thymine dimer, and that becomes a huge DNA adduct, a structural um, interruption in the path of the replication fork, and therefore becomes a challenge. Yeah. So these are the last two slides, I think. Yeah, we'll finish well in time. So other chemical mutagens also include base analogs. So what we discussed till now were the chemical alterations like depurination and deamination and the exposure to radiations like UV light. But also what can happen are the fact that base analogs can become a problem. What are they? Base analogs are bases that are very similar to the normal canonical nucleotide bases. They also exist in their normal state. However, they also have some rare tautomeric states or some altered conformations that are also present. So if 5-bromouracil, here abbreviated as 5-BU, gets incorporated in its normal state, what does it do? It is able to pair with adenine. What is the meaning of this? It means that when a cell is actively dividing, what it looks for are nucleotides. So under those circumstances, if instead of a normal DNTP being present, if there is 5-bromouracil which is present, or 5-BU, which acts as a base analog, then instead of the correct incorporation of the correct DNTP, base analogs can get incorporated into this newly synthesized structure. When that occurs, what it leads to is an abnormal base pairing that takes place. So what it does is 5-BU, when incorporated, this is 5-bromouracil, which behaves like thymine in its normal state, is able to hydrogen bond with adenine. However, in its rare state, what happens if it if it is in its rest state, it will pair with guanine in that condition. So what happens? What is the problem in this case? In the next round of replication, the 5-bromouracil G base pair gets resolved into a CG base pair instead of a TA base pair. So initially you would have expected TA to persist, but now it has changed to GC. Let's look at the schematic for more detail. So you have an AT to GC transition mutation. So the initial base pairing, the nascent base pairing is between T and A. When you add 5-bromouracil, it gets incorporated within this normal DNA. And at some point during replication, it switches to its rare or unusual state. When it does so, instead of behaving like thymine, it now starts behaving like cytosine, like you saw here in its rare state. When this happens, 5-bromouracil is now able to hydrogen bond with guanine. And in the subsequent generation, if again 5-bromouracil switches back to its normal state, then DNA replication will proceed. However, you will find that there has been a transition mutation and there has been a shift in the sequence of the bases. Alternatively, in a GC to AT transition mutation, we find the opposite of this. We find that there is a transition from CG to TA. So in both these cases, basically what you see is that there is a transition which has occurred from one type of base pairing into another. And also 5BU induced mutations can be reverted also by a second treatment of 5-bromouracil. But again, these are very dicey questions. We can never be completely certain which form or which tautomer it is currently in, and therefore exposure to these to these base analogs can become a problem. Finally, in the last slide, we are going to understand the function of intercalating agents. Now, as the name suggests, intercalating agents are those which bind or which integrate themselves into the DNA molecule. Examples being proflavin, acridin, ethidium bromide, the most common one, which is used to stain DNA for gel electrophoresis experiments. They are able to intercalate themselves between adjacent bases in one or both strands of the DNA double helix, as a result of which the helix is able to relax. If it inserts itself between adjacent base pairs of the DNA strand, which is the template for new DNA synthesis, an extra base can also get incorporated into the new DNA strand opposite the intercalating agent. So let's look here. This is the original DNA, the template, and the new DNA strand. This is the intercalating agent molecule which has inserted itself randomly between two bases at the time of DNA synthesis or DNA replication. So it gets any random base can get inserted opposite the intercalating agent. Here in this case it is G. What happens next is that when there is subsequent replication of the new strand, there has been a frame shift mutation because the base pair, a new base pair has been inserted, the frame completely shifts and there is mutation by addition. Contrariwise, there can be mutation by deletion as well, whereby the intercalating agent inserts itself such that there's again another round of replication where intercalating agent is lost and then it leads to loss of information. Let's go back to the legend. If the intercalating agent inserts itself into the new DNA strand in place of a nucleotide base, then when that DNA double helix replicates after the intercalating agent is lost, the result is a base pair deletion mutant. So there is a loss of the intercalating agent, and as a result of that, what happens is this is a subsequent round of replication. As a result of that, you will find information has been lost, and this is now known as a base pair deletion mutation. So these altogether can also lead to, I mean, there's an overlap between these types of mutations because, like you can see, the first one can be considered to be completely a frame shift mutation, whereas the second one can be classified under deletion mutations as well. So the range can vary depending upon the context. But yeah, intercalating agents overall can cause either addition or deletions. And therefore, frameshift mutations that you see here for intercalating agents, more often than not, they can be reversed by a second treatment with that same agent as well. However, of course, it's not foolproof, it cannot be assured, but it is definitely a possibility that does persist and is something that has been studied previously as well. So, with that, I think, yeah, we have arrived at the end of this class. Any points, any doubts, anything you want to discuss, please let me know. We'll discuss it. And of course, we'll do a recap of all these topics in the next week's session as well. So you can, compile your doubts and we can have the discussion later also. But if there's anything you want to discuss, please let me know. We'll go ahead with it. No? Okay, then I think I'll stop sharing the screen. The class has been recorded. I'll upload the recording as soon as I get access to the course portal. The previous class will also be uploaded there. But anyway, that terminates our discussion for today. Let me just end the slideshow. Yeah. So, following the flyer in the next class, we will introduce polymorphisms and the difference between mutations and polymorphisms, the topic of functional genomics very briefly, and then move on to sequencing methods, the traditional Sanger method, and uh, possibly in the week after that, we'll discuss NGS techniques as well. So, we'll take the course forward from there on. So thank you so much for joining. Please bring up your doubts. It's always a much better session when there's an interaction, when there's some contribution from both of us. So thank you so much for joining. I'll see you again in the next week on Tuesday at 6 p.m. as usual. I'll send a reminder email. In the meanwhile, once the course portal is available, you'll have the recordings available for you. Thank you so much. Have a good night.